Illumina sequencing is comprised of some basic steps shown here. In this video, we will walk through these steps and understand the basics of how this sequencing technology works. Our first step in the process is library preparation. Our library refers to the DNA we wish to sequence. Libraries can come in a variety of types, including from PCR products, which are commonly used for things like microbiome sequencing. But one of the real benefits of Illumina sequencing is its ability to tackle whole genomes. One of the biggest leaps forward for Illumina sequencing over Sanger sequencing was its ability to sequence multiple different sequences at once. Once your library is prepared via PCR or fragmenting, if you're loading a whole genome, we will add adapters and barcodes to each sequencing to each sequence in the library. Adapters in particular are critical and are short oligonucleotide sequences that are ligated onto the ends of each sequence in our library. These sequences are crucial because they allow our library DNA to stick to the flow cell, which we will see in the upcoming steps. Barcodes are used to identify unique samples. In cases where you want to sequence multiple samples at once, you can add unique barcode sequences to each sample. This then allows you to combine multiple samples in your sequencing run and then later separate them during data analysis. Once we have our library prepped, we can load it on a device called the flow cell. The flow cell is an amazing piece of technology that makes Illumina sequencing possible. Each flow cell is a lawn of short oligonucleotides that are complementary to those adapter sequences we added during library prep. So when we add our sample to the flow cell, it sticks to it following complementary base pairing rules. Once our samples are bound, we extend them, taking them from single-stranded DNA to double-stranded DNA using polymerase enzymes. Here we see our double-stranded DNA. Keep in mind as we step through here that this is happening millions of times on the flow cell all at once. We then remove our original sequence and leave a single-stranded DNA that is complementary to our original DNA. So why do we do this? One reason is that now our DNA strand is bound to the flow cell via covalent bonds, which is much stronger than our original hydrogen bond. Another reason has to do with bridge amplification, which is used to create little clusters of your sequence on the flow cell. On our flow cell are two types of oligonucleotide sequences. The first was complementary to the adapter sequence in our original sequence, and the second is complementary to the adapters in our newly synthesized DNA sequence, or said another way, was the same as our original adapter sequence. So now in bridge amplification, we bend the DNA over to the complementary oligos and synthesize using our polymerase enzymes. Then we denature this new double-stranded DNA, forming two single-stranded DNA templates, and then repeat the process. This keeps going with the goal of filling up our flow cell. Finally, we denature into single strands and cleave the reverse strands, leaving a cluster of forward strands on our flow cell. So here, near where each unique sequence originally bound, we have a cluster of same sequences, which will allow the sequence to be seen during our sequencing reaction. On, on our oligonucleotides, we now cap the 3' ends to avoid unwanted priming in the subsequent reaction. We are finally ready for sequencing. A primer that binds to our adapter sequence binds near the top of the sequence and begins to synthesize a new strand of DNA using special bases. Sequences are constructed and read one base at a time. The figure here shows how this process works. Each base in Illumina sequencing is a dd-NTP, which means it acts as a terminator base and has a fluorescent dye attached to it that allows it to be identified as an A, T, C, or G. So in a single cycle of Illumina sequencing, a single base binds to the growing complementary strands on the flow cell. In our example here, we see a G bound and it emits a green fluorescence, which is picked up by a hypersensitive camera. We can see at the bottom left of this figure in cycle 1, the G base is circled, and this is what the camera sees. Once the bases in a cycle are red, we can move on to the next step. The cool part of Illumina sequencing is that the terminator bases are reversible, and both the terminator and the fluorophore are removed before proceeding to the next cycle. Now we can flood the flow cell with our base mixture again and repeat the process. So if we look along the bottom of the figure here, we see the sequencer view of the process zoomed into a small area of the flow cell. So in each cycle, one base at each sequence location on the flow cell is determined, and the computer keeps track of the bases at each location on the flow cell. Essentially, it keeps a 2D map of the flow cell and allows us to assemble a potentially unique sequence at each location on the flow cell. The example here along the bottom of the along the bottom shows the assembly of 9 base pairs of a single sequence. But generally, this individual sequence will be at least 100 base pairs and as long as 600 base pairs. We zoom out a little bit, we see the scale of sequencing and can appreciate the awesomeness of this technology. Our sequencer is doing the process we just talked about up to 1.6 billion times in each cycle. The number of clusters is dependent on which version of Illumina sequencing you use, and there are several platforms. It's truly amazing when you think about the scale at which this technology operates. So in the end, what you get are many, many sequences output to a file that you can now submit to your appropriate data analysis pipeline. But often, before you do that, you need to do a step called demultiplexing. If you combine multiple samples into a single run, such as in microbiome sequencing, you can now use those unique barcodes to separate your individual samples back out into separate files in your analysis. The ability to multiplex and then demultiplex samples using these unique barcodes has made this technology affordable and allows us to sequence samples at a fraction of the cost that they would be. Demultiplexing is done entirely by bioinformatics software, which is actually often run on the sequencer during the sequencing run. We won't dig into bioinformatics any further here, though. The output of this sequencing technology is so much larger than that of Sanger sequencing, with even the smallest Illumina platforms generating orders of magnitude more data than this older technology. Illumina sequencing on the scale of genomes or whole microbial communities or whole transcriptomes is possible. And this has really revolutionized the field of genetics.